So I'll start off with a brief introduction on exosomes. So exosomes are essentially lipid vesicles that are secreted by the cells. These lipid vesicles are essentially characterized by their size, roughly around 40 to 160 nanometers. And what's special about these exosomes is that they're often correlated with various physiological responses. This is due to their ability to mediate short to long cellular communication. And exosomes are able to do this because they're heavily rich in various essentially metabolites, lipids, proteins, and nucleic acids that allows them to be directly involved in various physiological responses. So there has been an increase in interest in study exosomes for various diseases. And in our case, we're looking at using exosomes as biomarkers for cancer. So for the talk today, we'll be looking at a specific protein that is expressed in exosomes, specifically called PD-L1. So PD-L1 is essentially a protein biomarker that is highly expressed in various cancers. Their immediate function is to essentially suppress the immune system and therefore allow for tumor cells to survive. There are various forms of PD-L1. The more commonly studied one are the cellular forms of PD-L1, but more recently there have been papers that looked at soluble forms of PD-L1. There's monomer, dimers, microvesicles, and exosomes. And in general, there seems to be a stronger correlation between exosomal PD-L1 compared to other forms of PD-L1. So the challenging thing about looking at exosomal PD-L1 is that exosomes are actually very hard to essentially isolate and separate, and therefore it's also hard to analyze. Because of that, we generated new microfluidic technologies in order to essentially enrich these exosomes and also sort them based on their expression. So here is sort of a schematic of how our technology works. Essentially, we start off over here with our exosomes expressing PD-L1. We then conjugate them with antibodies targeting PD-L1, also conjugated to magnetic nanobeads. So essentially, exosomes with higher expressions of PD-L1 will essentially be more magnetic. And as we pass them through our chip is essentially layered with magnetic guides. So exosomes with higher level of magnetism will be able to withstand the higher deflection angles and be able to deflect further away from the center. So as they reach the outlets, essentially based on where or how far they deflect, we can essentially sort them based on their expression from high, medium, low. So the first part of our technology is essentially to enrich and sort exosomes. The second part of our technology is to use electrical chemical detection to essentially quantitate the amount of exosomes that are being captured or sorted. So in our lab, we've worked with various electrochemical systems that we've developed directly from our lab. One development was the nanostructure microelectrodes, where essentially we create these electrodes with very high surface area, allowing for high sensitivity and dynamic range. More recently, we've published a reagentless sensor technique that allows us to essentially detect systems at a very high detection time or very short detection time with no additional reagents needed. So here I just have two graphs just to illustrate the performance of our technology. We can see that we have a very high dynamic range ranging from 10 picogram per milliliter to 10 nanogram. That's what we've tested so far. Also, we can consistently detect this system at with essentially just 5 minutes. So to start, we looked at cellular and exosomal PD-L1 of various cell lines. And essentially, we honed down on these two cell lines here, H1975 and PC9. So first, we looked at the cellular levels of PD-L1. We're looking for 2 cell candidates for experiments with high expression and low expression of PD-L1. From there, we looked at their exosomes, and we could see that there's a significant difference between, a much more significant difference in their PD-L1 level in the exosomes compared to the cells. We see exactly the same results in our ELISA. This is just to double verify that our results are accurate. Once we've done that, we looked at our chip performance. So when we run it through our chip, there are essentially 2 parameters that we look at. One is the deflection efficiency. That's essentially telling us what percentage of exosomes are PD-L1 positive. And next is exosomal PD-L1 profiling. And this one tells us essentially the distribution of PD-L1 expression in the exosomes. So first, when we look at essentially deflection efficiency, we could see comparing our PC9 and H1975. Just a reminder, PC9 has low PD-L1. And from our deflection efficiency, we see that they're roughly the same. So that essentially means that the number of PD-L1 positive exosomes are essentially the same for both. When we compare it to our H1975 CRISPR knockout of PD-L1, we see that there's essentially negligible amount of PD-L1 positive exosomes. So this shows that our chip is very specific. Now that we looked at the deflection efficiency and concluded that they both have roughly the same proportions of PD-L1 positive exosomes, we then looked at the distribution of their PD-L1 in the exosomes. Red here is the PC9 and blue here is the H1975. And we see that the H1975 has much higher expression in exosomes or PD-L1 per exosomes. So this directly correlates well with our previous data here with our ELISA expression and our Western blot expression. So now that we have a working technology, we wanted to look at different applications. And one of the first applications that we looked at is essentially how tumor cells secrete exosomes after they're stimulated. So a common way to stimulate tumor cells and increase their PD-L1 expression is to add interferon gamma. And by doing this, we'll then monitor what type of exosomes are being secreted. Is there more PD-L1 positive exosomes, or is there a higher shift in the density of PD-L1 per exosome? So here is just a highlight of some of the data that we've collected. Essentially, our ELISA data shows us that there's a marked increase in exosomal PD-L1 for all cell lines that we've tested. When we look at the chip performance for H1975, we see that there's not only a higher proportion of PD-L1 positive exosomes, but there's also a higher shift in density for PD-L1 per exosome. So moving forward, there are actually a couple other validation experiments before we move on to in vivo and clinical studies. But essentially, we'll look at various immune cell effects based on different types of exosomes that we're exposing them to. And from there, with that data, we could essentially have a better success in our mouse study where we could validate how exosomal PD-L1 can modulate and affect immunotherapy. Finally, we've been collecting a lot of lung cancer patient plasma for months to years now. And soon, once we start a mouse study, we'll be looking at that as well. And here we'll be looking specifically at correlations with disease and also more specifically, where we're looking at responders versus non-responders to immunotherapy. So in summary of my presentation, there are 3 main points that I would like you to remember. The first is that exosomal PD-L1s are very valuable and understudied biomarker in cancer. Here in the Kelley lab, we have developed a microfluidic device that allows us to sort and profile exosomes based on their PD-L1 expression. And using this chip, we could learn a lot about the biology of cancer and how we could essentially help in the clinic. With that, I would like to thank everyone in the Kelley group, with special thanks to Kang Fu Chen and Alam Mahmood for their direct collaboration on this project.
Well, I'm curious, are the attached beads a problem at all for the downstream analysis of the sorted exosomes?
So essentially they can be a problem. That's why we've actually looked at bead release protocols. We actually have some data, but I can't actually access my slides right now for some reason. Essentially, we've looked at different bead release protocols. We've optimized it so that there's essentially significant release of beads while the exosomes are maintaining their shape and integrity. And from that, we're able to allow for downstream analysis without the beads.
Great, thank you. And I saw that Diana's hand was raised again. So Marshall, can you make sure she can unmute herself?
There we go. Thank you very much for that talk, Bill. Very, very interesting. I would like to hear your opinion about what the advantage would be of this approach in terms of cancer therapeutics compared to existing treatments. Do you have any thoughts about how this approach may actually be beneficial to cancer patients or doctors treating people with cancer?
Yeah, so this approach can be twofold, really. So one, it could help with screening. There's actually very little known about exosomal PD-L1 for screening, for diagnosis, and for prognosis. But another thing is that PD-L1 is actually a very established marker for immunotherapy. It's actually PD-1 therapy, actually. And there are actually been FDA-approved companion diagnostic where essentially people will measure the amount of PD-L1 inside the patient and then modulate the dosage of the immunotherapeutic agent before using it. That way it'll have higher success. And based on recent papers, it's shown that exosomal PD-L1 has a stronger correlation with the disease and the success of immunotherapy. So one of the biggest advantage of using this chip that I foresee is to able to better diagnostic for better success.
Thank you very much.